So I thought, what is the best way for me to teach some psychology without actually doing any prep? And I thought reading through a paper together might be the best way to do it. Psychology papers can be a little bit scary to look at in the first instance, but I think I should be able to walk you through it okay. So the paper we're going to be reading about is a Nature paper. So Nature is one of the top journals, or in theory anyway. There's a whole thing about how we classify journals, but a journal is where scientists publish their research. So it's basically just a fancy magazine, to be honest, except it's mostly online now. It is peer reviewed, which means that other scientists also have a look at what you've done, and they go through everything with a fine-tooth comb and make sure that it actually all makes sense. The paper we're going to be reading today is in Nature Human Behaviour, which is considered one of the more prestigious journals. It's if you can get your psychology published in Nature Human Behaviour, you're doing pretty good, you know. Your career is in science is going pretty well. The way that scientific research works is that you do your research and then you go away with your findings, you write them up into a nice little report, and you put them in a journal. So when I say a paper, I just mean a journal article. We kind of short form it to paper because it's just a bit easier to say. So this one that I'm going to be showing you all, actually, let me just bring it up. Let me bring it up. There we go. Look at that, a paper. So yeah, this is what a paper looks like. It is just a fancy formatted PDF with graphs and words and numbers. And a lot of people are absolutely terrified the first time they see these. Oh yes, there's also a long old list of references at the end. I'm going to walk through the paper. Bearing in mind, this is the first time that I have read this. I literally just read the title and I was like, yeah, that sounds interesting. So we're going to walk through it together. High replicability of newly discovered social behavioural findings is achievable. That's a lot of words. What does it mean? So, without having actually read the paper, which, you know, this is the normal way that you kind of find a paper, is that you don't necessarily read it, you just see the title and you're like, yeah, that sounds vaguely interesting, I might want to read that. And then you start reading it. Based on the title, I am assuming that this is telling us whether psychological findings, the results from psychological science experiments, can be replicated, means whether if you repeat them again, they kind of produce the same results. Even if you're doing it with a different set of people, you do exactly the same method and you see whether it produces the same results. Because if it produces the same results over and over again with different groups of people, then you can say, okay, this says something about humans as a group, right? But if it is instead, it has different results across different groups, then you can start to think, okay, maybe this is just kind of something that happens in a certain culture. Maybe it is something that happens at a certain time, or maybe it is just something wacky and we don't know what's going on. There's also some findings that can just happen by accident. So there's in any psychology experiment, depending on how you set up your experiment, there's about a 5% chance that you will just find what you're looking for when it doesn't exist. So that's just purely like a fact of statistics. Like it's just a fact of how random the universe is. Things just happen for some reason and they just occur. It's just a spontaneous thing. It happens sometimes. But it does mean that we have to try and work out the difference between an actual fact about how people work and a random occurrence that shouldn't have happened. So this string of numbers up here, every single paper has a unique set of numbers like this. It's called a digital object identifier, DOI, and it means that you can basically tell apart all of the studies just from this string of numbers. Now, that might seem really unhelpful and really random, but if you have got a massive library of literally hundreds, even thousands of papers that you're using for your research, you need to be able to just like have one set consistent link to each of them. So that's what these are for. It means that every paper that gets published has just a doi.org/ blah 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 URL. So I guess it's kind of like a link shortener because the links to the actual journal articles are always really, really long. And then next you've got information about when it was published. So you'll see here it says received, it says accepted, and it says published online. So basically, science papers don't get published as soon as they're received because they are all peer-reviewed. They are checked over by other scientists with expertise in the area. Because of that, there can be a little bit of a delay between getting the paper in at the desk of the editor of the journal and actually accepting it. So saying, yes, this is something that we are willing to publish and that we want to publish. So you can see here there was a bit of a gap of a few months. That's actually fairly quick. Sometimes they can take years. It really just depends on the individual journal. And then it also takes a little bit of time to get it published online because normally you just send it in as like a Word document type of thing, and then the journal goes through and edits it for you so that it looks all nice and fancy like this one does. The next thing that you're going to see on any journal article is just the list of people that were involved in the paper. So all of these people here were somehow involved in either doing the research or writing it up or making the graphs or thinking about how they were going to implement certain methodologies. So methodologies is just like the way that you do the experiment basically. And all of these people were involved. So this is a fairly big team, I think it's fair to say. You can get different types of scientific collaborations. You can get some of them, especially in psychology, they tend to be kind of smaller groups of people. So it might be like 2, 3, 4, 5 people working on a paper together. They should all have something to do with the research. And then all of these people have got these little numbers, so that corresponds with where they do their research. So you can see the first author here. So the first author is kind of the person that gets the majority of the credit, to be honest. If you're talking about a paper, so yeah, for example, this one, if we're talking about this one, we'd probably say Protzko and colleagues, because it's just a shorthand way of like referring to the paper. Because otherwise, if you're sitting here and you're like, John Protzko, John Krosnick, Leif Nielsen, etc., etc. It goes on and on and on, especially if you've got a really big collaboration. If you think about things like CERN, there's like thousands upon thousands of people that work on those experiments. It's not as common for that to happen in psychology, but it's starting to happen a little bit. So yeah, once it gets to the point of like more than 5, you start to get really tired listing off all the names. Although it's kind of rubbish because all of these people just become and colleagues, which is like their work is kind of being distilled into a pretty short set of words, which is a bit sad, but you know, it is what it is. Yeah, everybody gets at least one first author paper, hopefully, so you know, hopefully they will get their time. And then this bit here is called the abstract. So this is just basically a summary of the whole paper. It's condensing the entire paper, so all of the research, what they found, and all of their conclusions into about 200 words. But yeah, the 200 words, it's actually really hard to kind of think about this massive set of experiments that you've done and try and squish it all down. This paper reports an investigation by 4 coordinated laboratories in the prospective replicability of 16 novel experimental findings using rigor enhancing practices. That basically just means there's a whole bunch of scientists that got together. There are 4 different labs. So that means a lab is just like a group, well, it's short for laboratory. But in psychology, we don't always have actual physical laboratories. We sometimes just kind of form a group of our mates and say it's a lab. So there's 4 labs that have come together. They all work on similar topics and they've thought, okay, let's test some things and see whether this process that we're using actually works and whether new ways of doing science kind of make things a bit more consistent. The rigor enhancing practices that they used are confirmatory tests, large sample sizes, pre-registration, and methodological transparency. That's a lot of fancy words, isn't it? So, confirmatory tests. That is just a specific kind of statistical method that we use on the data that we've generated. And that's it. That's all it is. It's just a specific type of statistical analysis. Large sample sizes. So that literally just means that we are performing our experiment on a big group of people, as in thousands. Thousands are ideal, because the more people that you have in your sample, the less likely you are to generate findings that they're not consistent across groups. Because if you've got a really big sample, then chances are you've got a fair number of groups in there, right? Pre-registration is when you basically declare in advance what you're going to do. So you sit there and you write out a little thingy that says, hey, I'm Psydere, I want to run this experiment about I am testing anime fans. I'm gonna test anime fans. I'm gonna put them in a room, and then I'm gonna make them interact with somebody who is or isn't an anime fan, and then this is how I'm gonna analyze it. These are the statistics I'll use. And that's all pre-registration is. It's just declaring those things up front so that you can't go away and faff around with things and cause problem. One of the things that really annoys me about scientists is that we have this tendency to just use jargon that just isn't needed. We just don't need to do it. We just use these words to make ourselves feel smart, and the reality is that scientists are people like me. Like we're just normal. Let's put it that way. We're just normal. But yeah, it's not needed. I think people would trust science a lot more if we didn't use these kinds of words to make people feel alienated from it. Although funnily enough, as I was talking, I've just realised alienated is also kind of a fancy word, so I'm not really practicing what I preach, am I? In contrast to past systematic replication efforts that reported replication rates averaging 50%, replication attempts here produced the expected effects with significance testing in 86% of attempts, slightly exceeding the maximum expected replicability based on observed effect sizes and sample sizes. They really like to make their sentences long and difficult to pronounce. Remember when I said that sometimes psychology, we run experiments and it doesn't do what we expect it to do always. Sometimes it will not repeat, so we will not be able to repeat the experiment and find the same results. So that's what it's referring to. There were some studies that came out, I think kind of around 2013 time, which showed that there was about, if you were going to repeat a study like that, you were going to replicate it, then you would have about a 50% chance of it showing the same results and about a 50% chance of it showing something totally different, which is not ideal, because for us to be doing proper science, we kind of just want it to be like closer to 100. Like we kind of want to be doing science the right way the first time. And if we're finding consistent patterns, then we should be seeing it all the time. We shouldn't be just randomly finding some things replicate and some things don't. So yeah, those original studies found that, but the way that science is done, especially in psychology, has totally changed in the last kind of 10 years. It's shifted much more towards being really careful about how we plan out our study. And it's also shifted a lot more to using systems that stop us from doing research in a way that isn't best for finding consistent results. So what that means is that the average rate of replication has gone up from 50 to 86%, which is a good thing. It means that the way that we're doing science is genuinely improving. It also means that we're not all the way there yet, because we use this p less than 0.05 cutoff. So that is literally just we expect about a 5% chance that we're going to find something when it doesn't exist. So ideally we need this 86 number to be, we need it to be 95, but it is not there yet. So yeah, we've still got some work to go. When one lab attempted to replicate an effect discovered by another lab, the effect size in the replication was 97%, that in the original study. So what is an effect size? An effect size is basically a number that we use to kind of understand how big the effect is that we're looking at. If we're taking that example again of putting an anime fan in a room and then introducing them to another anime fan or somebody that doesn't like anime, and then we're expecting there to be a difference in how they interact, let's say just how friendly they are, then we would describe the difference between those two experimental conditions as being the effect size. So if one group, let's say if there was 2 anime fans together and they interact for 50% more time than in the other group, then we would say that the effect size is 50% bigger. So the way that papers tend to be structured after this introductory bit with the information about publishing, the title, the authors, and the abstract, you'll jump straight into the introduction section. Now, the whole point of the introduction section is to kind of give you a broad overview of the thing that the paper is about. So in this case, the paper is about replication rates. The introduction is going to be giving us a bit of background about why replication rates are important, how they've changed over time. This is just an example of some of the effects that they're looking at. Because this is a nature paper, they do things in the wrong order. What's meant to come after the introduction is the methodology, but for some reason in nature papers they put it at the bottom. So the methodology is telling us how they actually conducted the research, what they did. And to be honest, I think this is probably one of the more important bits of a paper. I mean, they're all important in their own way, but this is the bit that you need to look at to really understand what they were doing and why they did it. So it will tell you things like the people that they decided to recruit for the experiment, and it will tell you about what they actually did. There should be a kind of step-by-step guide to doing it. And then that is followed on by the results. So this is just a whole bunch of statistical tests. Normally you get some lovely little graphs in here. And there should be the occasional explanation for what all of these numbers mean, but not always. And then once you're done with that, you get to the discussion. So if you struggle with numbers, with statistics, I would always suggest for people to go to the discussion first, read through it so you understand what they think they found, and then go through and have a look at the numbers and see if they match up. Low replicability can be the consequence of false positives or exaggerated effect sizes among the original studies, resulting from low statistical power, measurement problems, errors, p-hacking, and regression to the mean due to selective reporting favouring original positive results. Now, that is a whole bunch of things which we're not going to get into today. But what I will say is that historically, science hasn't necessarily favoured doing good science. So what do I mean by that? The system that scientists work in is very numbers orientated. So universities are really looking for things like how many papers are you publishing? How many people are referencing your research? How many people, foundations and government organizations are giving you money to keep getting your research done, especially in psychology, where we can churn out like surveys and kind of mishmash ad hoc experiment very, very quickly. And if you get a data set and then you find that you weren't able to get the finding that you wanted, you can just kind of do some different statistics on it and you'll probably find something, even if it's not what you were looking for. Now, a lot of the time, that's just, remember I said earlier, sometimes things just happen randomly. Scientists will keep using their old data sets and doing new analyses on it, even if they really shouldn't. And they will write new papers from it. And all of this has come together and is the reason why psychology doesn't replicate very well. Different analytic strategies, what that's referring to is just the way that different scientists will run their analyses, just purely down to how they've been trained or just the norms in their field, things like that. They understand the numbers in different ways, just slightly different, but different enough for it to sometimes have an impact. And yeah, that just means that sometimes things seem to replicate when they shouldn't, or seem to not replicate when they should. So, yeah, this is just kind of highlighting the fact that yeah, sometimes things don't replicate just purely because of people are diverse, and scientists are fairly diverse too. And sometimes people are they're doing things in different ways, and that's okay. It just happens sometimes. Failures to replicate or declining effect sizes can be the consequence of unknown or unarticulated moderating influences and boundary conditions that differ between the original and replication studies, indicating shortcoming in the theoretical and methodological specification. So we have these ideas about, we have a theory We have a theory about how anime fans interact, but we have some kind of limit to what we think our theory can explain. So that's basically what a boundary condition is. It's really important when we're doing science to work out the boundary conditions in our theory, because that's telling us where the theory stops being relevant, where we can stop applying it. And it also tells us the limitations that we might see in our methodology. All of these are things that affect whether our effects appear and whether they are the same size when we try and replicate them. In this article, we report the results of a prospective replication study examining whether low replicability and declining effect sizes are inevitable when using proposed rigor enhancing practices. Do you remember I said earlier about how we're trying to think of ways to do science better? So for example, by telling people in advance what kind of statistical analysis we're going to do. So that's exactly what it means. They are examining whether these rigor enhancing practices like pre-registration have an effect on replicability and effect sizes. This wasn't just psychologists. A lot of the things that I've talked about so far and that they're talking about in this paper, they're not just issues in psychology. They are issues across all of the social sciences. So yeah, everything from marketing to political science, to judgment and decision making, so understanding how people think about things. I think they are also a problem in economics a little bit, but not as much, because economics is a little bit more mathsy and they use big samples by default. So they tend to do like kind of countrywide analyses, more focused on the economy. Shock and horror. Economics focuses on the economy. I can't believe it. Each lab engaged in a pilot testing of new effects based on their laboratory's business as usual practices. These could involve collecting data with different sample providers and with any sample size the lab saw fit. All pilots were required to have their materials, procedure, hypothesis, analysis plan, and exclusions pre-registered prior to data collection. Ostensible discoveries were then nominated for confirmation and replication by the discovering labs. The main criterion for moving from piloting and exploration into confirmation and replication protocol was that the lab believed that they had discovered a new effect that was statistically distinguishable from zero during their piloting phase. So in short, they're taking their kind of initial ideas. So a pilot test is basically when you have this idea for a study, but you're not entirely sure it's going to do what you think. So you take it away, you run it with like a smaller group of people. It depends on the study. It might be, like 20 to 50. It might be a few hundred if you're trying to do a large scale study. But the whole point is to test whether what you're doing is actually going to work, or whether there's any changes that you need to make to ensure that it's actually doing what we think it should do. So the idea here is that they're doing these pilot testing like as a collective, right? They're kind of a big group of labs. They're all coming together. They're reporting the pilot testing ideas that seem to actually do something, and then they are taking those ideas and doing a bigger confirmatory test, trying to make sure that it's actually worth doing this study with a big sample, and then whether we actually find it when we do. From just reading this initially, this seems like a really cool idea, because one of the big problems that we have in social sciences generally is that we don't really get that much money. We don't get much money to conduct our research, which we do need. It's not ideal trying to run an experiment. Say, for example, you're bringing people into the lab and they have to sit down and do like a 2.5 hour study where they're just sitting and pressing buttons on a keyboard, and then you turn around to them at the end and you're like, okay, thanks for taking part, and that's it. You don't get anything from it apart from like the satisfaction of helping science, I guess. But it's a lot of people's time and a lot of people's energy and effort. So yeah, we should really be compensating them. Each of the 4 labs submitted 4 new candidate discoveries for a self-confirmatory test and 4 replications, for a total of 16 confirmatory tests and 64 replications. What was happening here is that the labs, they've all done their pilot testing. They are running their nice little introductory study to see whether there's an effect size that they want to check, and then they are replicating it themselves. So the first lab has done a small test, and then they've done a big test. And then they give it to 4 other labs So 4 groups of scientists that, you know, then they're doing their own thing. They're nothing to do with the original lab. And then those labs repeat it in exactly the same way that they did it. So it ends up that there are a series of pilot discoveries, and then all of them are replicated at least once, and then if there is still an effect being found, they are repeating them again to make sure that they are actually an effect worth studying in the future. Oh yes, and they used a large sample. So remember I said earlier that it's important to have a big sample. So that just means a big group of people that you're doing your research on, so that you can start to see whether it's something that you might expect to see on a big scale across the whole population. Regardless of the outcome of the self-confirmatory test, in the replication phase, all 4 labs conducted independent pre-registered replications using the written methodology and any specialised study materials shared by the discovering lab, for example, videos constructed for delivering interventions. So yeah, whether the replication that the first lab did found an effect size that matched the pilot or not, they still just ran it again just to make sure. Sometimes you can actually find things that don't really exist, but you can also not find things that do exist. And again, that is just a function of randomness. Strong communication between labs to maximise sharing of tacit knowledge about the methodology, but in this case, to maintain the independence of each replication, we opted to discourage communication with the discovering lab outside of the documented protocols, except for critical methodology clarifications. So, that means that normally collaborating, so, you know, all the scientists coming together and working on the same thing, we love that, we support it. It makes science more effective and better. But in this case, we are testing whether the changes to the scientific process, such as pre-registration, actually end up creating better results. So because of that, we can't be having people collaborating through back channels. We can't let people talk. We can't let them share things about the studies that might have an impact on the way that the replicators are conducting the research. If there is a bunch of these replications happening where people are talking about it and they're sharing their knowledge about how to do the study, and then there's a bunch of them where they're not, then that means that you're going to get probably quite different results across each. So you need to make sure that nobody's talking. We need to control for it, you know. That's the word. We're controlling it. It's a variable that we are controlling. It means that we're not letting it change because it could affect the results if it does. The replicating labs used equally large sample sizes, and each lab used a different sample provider. So yeah, they're all conducting the studies with the same large 1,500 people. Group of people, but they're all using a different service to recruit their participants. So remember when I said that we need money in order to do the best science? That's what they mean. They are using some kind of service that pays people for taking part in the research, and each one is using a different one. And I think the idea there is that if there's any differences between them, they'll kind of even out because everybody's replicating multiple studies. Pre-registration, reporting all outcomes, large sample sizes, transparent archiving, sharing of materials, and commitment to high fidelity replication procedures should minimise irreplicability or declining effect sizes stemming from questionable research practices, selective reporting, low-powered research, or poorly implemented replication procedures. In theory, all of these changes to the scientific method should mean that we are replicating things consistently. So the irreplicability, so that just means inability to replicate findings, to find them again and again, and declining effect sizes. So remember, the effect size is getting smaller each time you test it. It means that it should, in theory, according to what we know about the scientific method, hopefully mean that we're finding things more consistently. Question: in psychology, if you make the sample size quite large, are we risking introducing groupthink into the experiment? Like the results being skewed by the participants being aware that the study is large? So that's a really interesting point, because I know that there is a lot of research coming out around this phenomenon called the wisdom of the crowd. So wisdom of the crowd is this idea that if you get lots of people together and they kind of estimate something, they kind of make their best guess about how likely something is to happen, you get much more consistent and accurate results than you do if you just have a few people, or even people that are experts in the thing that they're meant to be guessing. So because that's becoming more common knowledge, in theory, that could become a thing that influences the experiment. So what you're saying there about participants knowing what the study is doing, and then that, or something about the study, and then that influencing how they behave, is called a demand effect. And it's something that we have to think about really, really carefully in psychology. I think that's actually a thing that I have never seen mentioned before. But yeah, so you might have actually just come up with your own unique experiment there. I think there is a chance it could happen. But I also think that if there were any evidence to suggest it was likely to happen, we would probably just not tell people how many we're aiming to recruit into the experiment. So I think I've mentioned in a previous stream, but when you're working with people, you've got to stick by some really, really strict ethical rules. And that means that for the most part, you have to be really upfront about what you're doing with them. But you can hide certain things from your participants as long as you are upfront with them at the end. You tell them everything that you did, and you tell them exactly why, and you give them the chance to remove their data from the study if they get to the end and they're like, nah, this isn't, I'm not about this. This was sus. I am not about this. So yeah, if you give them the chance to kind of opt out and remove their data at the end, then you can hide it from them. So we've got through that bit. If despite these rigor enhancing practices, low replicability rates or declining effect sizes are observed, such rates or declines could be intrinsic to social behavioural scientific investigation. So this is basically just saying, if we're trying all of these new ways of doing science that are better in theory, and the results we're getting still don't replicate, then we need to seriously consider whether social behavioural scientific investigation, so basically just social sciences, psychology, political science, all of those fields, we just need to consider whether they're a bit rubbish. We just need to think about whether we could just, we might need to just rethink the whole thing. But as we know from the abstract, things are a little bit more positive than that, and things do seem to be okay if you've got a good system in place. So that's really good. Each of the 16 ostensible discoveries were obtained through pilot and exploratory research. So exploratory research is just research that's kind of like putting feelers out and seeing what new ideas there are. Not every pilot study the labs conducted was put forward for confirmation and replication. Like all exploratory research, labs sometimes found errors, did not find signals of potential effects, or just lost interest in pursuing it further. Yeah, sometimes we start researching something and then we get bored halfway through, and that's okay. It's okay. Normalize giving up on ideas that are rubbish. The labs introduced 4 provisional discoveries each, resulting in 16 self-confirmatory tests and 64 replications, so 3 independent and 1 self-replication for each, testing the replicability and decline. All confirmatory tests, replications and analyses were pre-registered both in the individual studies and for the meta-project. That means that they did all of the fancy new science things for both the individual studies run by the labs, and then also when they were pooling everything together, because this is a paper where they're taking all of those studies and squishing them all together, they've also pre-registered what they're going to do for the big squish. Nature papers are strange, and they put the actual methods at the bottom. So we've got some graphs here. Size is S, lab is a different shape. Okay, so the different shapes represent different labs. So there's the confirmatory test, so the first test that the original lab did. They did their pilot tests, and then they did their confirmatory tests, and then they replicated their confirmatory test. So the same lab does the same study, they just replicate it exactly as they did the first time. And then they replicate with other labs. So other groups of scientists come in and have a little attempt at running the study themselves, and they see what they can do. And it looks as though, because these are all meant to be replications, we're expecting them to all be in roughly the same place. So that means just like on the x-axis, this one at the bottom here, they should be in roughly parallel. So for example, you can see here that these ones, they all kind of roughly cluster around this line between 0.3 and 0.4. And that means that they are finding a somewhat consistent effect. You can't really tell just from looking at it. You have to ideally use stats to like work out whether it actually means anything. So just making plots like this is a really big part of interpreting results. You can tell just by the symbols, the confirmatory test yellow is first and the replication blue has the same symbol. Oh yes, they do. And then these ones, the green ones, are different labs, so they've got different symbols. So just from having a very quick look at these, it seems like the majority of them do roughly replicate in the way that's expected. I would say the ones that are really sticking out for me are Redemption, Lab 2, so second row down, last one, last column. So this Redemption one here, the bar going down, the black bar going down the middle is the 0 point. So that means that the original lab found an effect going in one direction, and the second, like the replication lab. So all of these replications over here, they're on a different side. So that means that the effect that the replications found is literally the opposite of what the original studies found. So that's a bit weird. And then if we look over here, this one in the bottom left, they're all just kind of spread out. Like there's quite a diversity in effect sizes. Like 3 of them are on 0.2. One of them is around 0.1. And then one of them's around 0.45. So what's going on there? That's kind of sus. And then again, lab 3 over here, that's even wackier. Like they have found everything from 0.3 to 0.6, which is like a massive effect size. So yeah, there's some interesting stuff going here, but it does seem like on first glance, most of them are fairly consistent. So that's good, that's good. That means that the replication method is doing what we expect it to do. Oh yeah, also, here's a funky little thing for you to know. When there's little bars coming off the side like this, that indicates a confidence interval. So a confidence interval just tells us roughly we have a 95% chance of the effect that we're looking for being within these lines. And this is what we think it is on average. Just putting all of the data together, that's our best guess, but it could be anywhere between here. Now, if it's all consistently on one side of this 0 line, that's great, you know, that means that we're consistently finding effect and we're confident we've got an effect. But this one here is crossing over the line, which means it could be that there's an effect, but it could be that there's no effect. So that we would generally consider to not be a significant finding. So I would always say, look at the graphs before you go and look at the numbers, because stats are wacky. And sometimes you read it, and even if you know it, like I know stats, I teach stats as a job, I know how they work, I, you know, do data analysis as part of my PhD, and I still sometimes look at like the way that papers report stats, and I'm like, what is that? Like, I cannot process whatever it is that you are doing right now. So I would say always, if there is a graph, which hopefully there is, there should be some kind of data visualization, because they are super important for, you know, just getting to grips with what's going on. But always go look at that first. And then once you think you can work out what's going on in the graph, go away and then go and have a look at the stats and see if they match up with what you expect from the graph. So all of the graphs should have like a caption thing at the bottom, which tells you roughly what they're doing. The best graphs, you just read the caption and then you just instantly know what's going on. Sometimes you read the caption and you're like, I still don't know what's going on. It kind of varies. Difference in effect size estimates between self and independent replications for 16 new discoveries in the social behavioural sciences compared with the initial confirmation study effect size estimates. Differences in effect sizes between self-replications and independent replications for the 16 discoveries compared with the self-discovery test effect sizes. Okay, so what this graph is doing is they've set the original effect size that they found as a reminder. An effect size is basically just a way to quantify the difference in size between your experimental conditions. So they've set the original effect size that they found to 0. And then what this graph is showing is how much each of the replications varied from that original effect size. So you can see here that some of them got smaller and some of them got a bit bigger. This one especially got bigger. This is the one up here that looked wacky as well. Some of the replications were bigger, and then you can see exactly the same thing on the same graph, or on the other graph even. You can see exactly the same thing on the other graph. So we like that. That means that our graphs add up. They are telling us different aspects of the same thing. And that is always very nice because it means that you can get an understanding of different aspects of this paper's findings without even having to read the findings yet. You know, we're getting an idea of what's going on just by looking at these. So we love that. We love that. Please, if you ever do like your own research, put in some good data visualization. It makes all the difference. It makes so much difference, honestly. Discussion sections basically take the results and they put them into context by explaining them. So they kind of link them back to existing literature. They say, this is what we found and this is potentially what it means. So it's a really important section for just trying to understand what the authors think about their own research. It's the place where authors of papers can kind of sell their thoughts. They can sell their theoretical contributions. They can sell why their research is super important. And that is pretty cool. But it does mean that you need to check at least the data visualizations before you dive straight into the discussion. Because this is the bit where they can just share what they're thinking. Rather than beginning with published findings and attempting to replicate them in a retrospective replication investigation. Wow, retrospective replication, that's a tongue twister. Retrospective replication investigation. We implemented a prospective investigation. By discovering new experimental effects, we were able to apply a meta-scientific lens to the entire process from discovery to a confirmatory test and through a sequence of replication attempts. So that's just basic them saying, like, look at us. We did things right the whole way through. We started off with a new way of finding new findings, and then we applied all of the existing funky methods we've got to make robust findings after we found those. So yeah, they're doing the whole process, the whole pipeline in this method, you know, from start to finish. By subjecting ostensible discoveries to large sample pre-registered confirmatory tests, we ensured that the primary findings for replication were free from p-hacking and questionable research practices, unlikely to be artifacts of low statistical power, and fully documented. So yeah, that's just them bigging up their funky, you know, newfangled open science methodologies, you know, which is good. We love that. We love that. It means that they're making probably better findings than I ever will, to be frank. Yeah, they're using the large sample because that makes it more representative. So it's more likely to kind of, you know, be something that we find consistently in the population. It is pre-registered, so they've put their analyses online before they did them to kind of minimize the chance that they would go away and do something dodgy with the data. Because believe it or not, that's a thing that's happened. Oh, pre-registered means the methodology is registered in advance or the data. So it's the methodology and the way that you're analyzing the findings. So normally you would do it before the data's been collected. But there are some situations where you can pre-register just analyses for an existing data set, but it should really be a data set that you haven't seen before. Yeah, the whole thing that caused the need for pre-registration, or rather, you know, led us to a point where we start using pre-registration regularly. We found this thing called p-hacking and also p-phishing. So p-hacking is where you have a data set and you run the tests that you expected to run, and they aren't quite significant. So when you're running statistical tests, you're looking for like if there's a difference, and if that difference is meaningful, we judge it with a kind of arbitrary cutoff, to be honest, but it's just how likely is it that we could find this by chance, and we go with a kind of standard threshold of 5% chance that it might just appear randomly and 95% chance that if it's there, then it's actually there. Pre-registration is meant to kind of guard against that because you're saying in advance what statistical tests you're going to run. So if your findings aren't statistically significant, you can't go away and then run a different test and then pretend that you were going to do that the whole time, and you can't go away and, like, you know, exclude certain individual bits of data to try and make it significant when you run the test. And fully documented, yes, we love fully documented research. We love fully documented anything. It means exactly the same thing as it does with like coding stuff, so like programming and you know, software development and stuff. You know, you just keep track of what you're doing and how it works and what to do if anything breaks. So for example, with my data sets, I will set up what I call a codebook, and it is basically just a big old PDF which lists out every single variable in my data set, and it says what the questionnaire item was, it says how it was measured, it says what questionnaire overall is meant to be part of. As a programmer, I also love fully documented things. Yes, you know what's up. Honestly, there's nothing worse when you like download, well, yeah, in my case, when I download a data set and I'm like, oh yeah, I can use this for something, and then there's like nothing. Like, it's just a CSV file plonked on the internet, and the variables are all like CZA73, and I'm like, what is that? Like, and there's no questionnaire linked to it, so I can't even work it out. Honestly, it's the worst. And yeah, I've seen terribly documented code in my time as well, and that is also depressing. Yeah, it's painful. It's painful, it's depressing, it makes me angry. Okay, right. These 16 discoveries, 4 from each of 4 independent labs, were then subjected to independent sequential replication, 55 out of 64, or 86 supported at p less than 0.05. So that's the arbitrary 5% cutoff that I said about. And then D, so that's an effect size, is 0.26. I think that Cohen's D is roughly telling you the proportion of variance that can be explained by your experimental manipulation. But in this case, it's all of them, I would assume. Like, they've done some kind of meta-analysis to like smush them all together into one statistic. Considering replications of only the 13 statistically significant self-confirmatory tests, the observed replicability rate was 90%. That's good. We love that, because frankly, up until this, the replication rate in psychology at least is like 50%, if that. And things that like are actively being researched in a lab cannot be replicated within the lab. So yeah, it's not ideal. But this seems like a really promising way to counter that, right? If we can start bringing labs together to do independent replications of each other's work, that'd be great. Like, I would love to do replications of other psychologists' work. It sounds really fun. Okay, right. So, considering the power to detect effect sizes from all 16 self-confirmatory tests, the replication rate could not be any higher. Well, that's fair enough. I mean, 90% is pretty good. Prior replication efforts in the social behavioural sciences report replication success rates of about 50% on average. Oh, yeah, that's what I said earlier. Maybe I got that from this paper. Producing effect sizes less than half the originally reported effect sizes. So yeah, generally if you test the same thing again, you're going to find an effect size that's smaller. Sometimes it might just be because the person replicating did the experiment differently to the original researcher. Sometimes it's just a phenomenon called regression to the mean. So if you do things over and over and over again, if you start off really, really well, eventually chances are it's going to kind of like come down to an average point that's like in the middle, which is why there's like this thing about sometimes you can like peak too early. It's because if you get really good success early on, then there's a chance you're just going to regress to the mean. It's not necessarily a reflection on you. Sometimes it's just statistical chance. The effect sizes overall were generally the same, or fairly similar, which is great. Yeah, because honestly, like if you repeat things over and over and over again, the pattern seems to be that they go down. I should also mention, actually, there could be sometimes there's a feature of just like time. So for example, there's a few experimental manipulations that were used over and over in the 90s that just don't work anymore because people don't understand it, or it's just not a thing in society anymore. If you replicated a study from the 90s that was like dependent on sending a fax, I mean, I'm sure there's a lot of people alive now that haven't even seen a fax machine, which is upsetting. The present findings establish a benchmark showing that high replicability in the social behavioural sciences is achievable in both statistical inference and effect size estimation when the original studies and replications are conducted using ostensibly best practices and replications are conducted with complete fidelity. If you do research in a way that, you know, actually makes sense and is actually consistent and robust, surprise, surprise, can you believe it? You get good findings. You get replicable good findings. Like, wild. I can't believe it. That seems like a fair enough conclusion based on the very cursory look that we had at the data visualization. We investigated whether low replicability and declining effect sizes should be expected from the social behavioural sciences because of the complexity of the phenomena, hidden moderators, and other factors that might be intrinsic to the phenomena being studied or to the replication process. Social sciences, they are a lot more complex than people give them credit for. There's a bit of a meme that, you know, like psychology and sociology and all of that are like soft science. I don't know what to say. There's a lot more factors that you need to think about sometimes. It is not just sitting down and asking people how they're feeling. I mean, sometimes it is. Sometimes it is. But that is not the majority of social sciences these days. A lot of social sciences actually involves really complicated things like network analysis and stuff like that. I'm not even going to try and explain what network analysis is. It's just looking at like social networks, but like applying pretty intense statistical models to them. Turns out there's actually a whole bunch of like complicated stuff hidden in there. You need to think about things like social context. Is this phenomena that we're looking at, is it a product of the time or the place? You know, there's a lot of research into social media, how people interact with social media, how people interact on social media, how social media interacts with the systems that we live in. And that is not easy to research. And it's also a whole area of research that didn't even exist like 10 years ago, because 10 years ago, the social networks were still fairly new and people were only just starting to research them. Wait, 10 years ago? That was 2013. Damn, they weren't that new then, actually. Okay, so like 15, 20 years ago, this wasn't this wasn't really a research area. But it is now. It's actually, I think that from what I've seen, social media seems to be a pretty substantial chunk of social psychology and, well, social sciences in general, to be fair. It's becoming a big part of it because a lot of us are living really big chunks of our lives digitally at this point. Even you lot, even us right now, we're living our lives digitally. Isn't that wild? People thought that maybe the lack of replication is just like because social sciences are so wacky and they've got so many degrees of freedom and they're just too complicated. And also because they're time bound and culture bound a lot of the time, or they vary according to time or vary according to culture or to generation, things like that. So they thought maybe we're just not meant to have a good replication rate. Maybe we are just to be banished to the soft sciences forever. Yeah, this paper seems to show the opposite, actually. It seems to show that we can have replicable findings and we can have consistent, robust effects that actually mean something. So that's exciting. The present results are reassuring about the effectiveness of what we think as best practices in scientific investigations. All of those things like pre-registration, sharing things openly, all of that jazz. It seems like it does actually work in the way that we expect it to. It does actually seem to improve the scientific process, which is really good for psychology and social sciences. But it's also just really good for science in general, because it's not just us that can use this. Every scientific domain can use this. We're developing how we do science to make it more robust, and I think that's really cool. When novel findings were transparently subjected to pre-registered large sample confirmatory tests, and when replications involved similar materials and were implemented with a commitment to faithfulness to testing the same hypothesis with fidelity to the original procedure, the observed rate of replication was high. So yeah, if you pre-register your analysis plan, if you pre-register your methodology, and then you do the science in a good way with a large sample size and transparently share things, and then if replications properly involve going through exactly the same procedure as the original studies, then it turns out It worked. Furthermore, we saw no statistically significant evidence of declining effect sizes over replications, either when holding materials, procedures, sample size constant, except for sampling error, or when materials, procedures, and sample sources varied but were faithful to the original studies. So no matter the type of replication, if it was exact or if it was just slightly different, further showed with one survey. Ooh, they did surveys. Nice. We further showed, with one survey of scientists and one survey of the lay public, that our studies were not a priori more predictable than similar other studies with a known replication rate. I guess they must have asked whether people thought things were going to be replicable. Oh, there's an entire graph I missed. Oh, okay, so that just shows how the effect sizes were changing over time with each replication. So an uninteresting reason for high replicability would be if the discoveries, although novel, are obviously true. Trivial findings might be particularly easy to replicate. To assess this, we conducted 2 additional studies at the University of California. In the first study, 72 researchers reviewed a synopsis of most of the research designs and predicted the direction of each finding. So if you think about an experimental design where you've got 2 groups, and we would be testing whether one group increases in their hours of anime watched compared to the second. I don't know what we're manipulating. Exposure to VTubers. We're researching anime fans. Disgusting. So how are we researching anime fans? We want to see if, our hypothesis is just basically a statement which says what we're expecting from our research. So we're gonna say that our hypothesis is we want to see if exposure to VTubers makes people watch more anime, or does it make them watch less anime? To make an experiment like that work, we need to just pick one. So the increased anime or decreased anime, those are 2 different directions of the same effect. A hypothesis needs 2 aspects. You need to know what you're changing, your independent variable, and you need to know what you're measuring, so your dependent variable, because the dependent variable is dependent on the changes that happen in the independent. So our IV, exposure to VTubers. So that means that we have 2 groups. Our first group is people that are shown let's say they're shown a 15-minute clip of a VTuber. We've got group 2. When you're running an experiment, you want to make sure that your different conditions of your experiment, the different things that you're testing, are actually comparable. So if you've got something like a 15-minute clip of a VTuber, you also want to make sure that you can actually compare it to something meaningful, right? You also want to see whether anime would do anything different to VTubers. So we're going to have group 2 be shown a 15-minute clip of anime, just like a standard anime program can be whatever. And then group 3 is our control. So that means it's the baseline group. They're not shown anything. If we want to be really, really thorough, we can actually also have a group which is shown a 15-minute clip of real people. So we've got our 4 groups. This is actually a pretty complicated experimental design already. So just for the sake of this, I'm gonna get rid of these. I want to make it simpler so that you can understand this more clearly. So we've got our two groups. We've got the anime fans. We've brought them into the lab. Some of them, half of them, we're showing them a 15-minute clip of a VTuber. Whatever VTuber you can choose. It doesn't have to be me. I'm fine if it's somebody else. I won't judge. You have a second group. The other half are shown no clip at all. So they just sit in a room and they just twiddle their thumbs and vibe. And then DV is what we're measuring, because remember, we're seeing if the change in this is dependent on this. So that's going to be number of hours of anime watched in let's go with a 7-day period. This is very easy to measure. We're just going to ask them. Or if you want to be more thorough, sometimes people lie. They'll say, yeah, man, I haven't watched any anime. And then they've like binged watched the entire of One Piece in a week somehow. Like they've literally bent time and space to do it. And other people, they'll be like, yeah, I've watched loads of anime. And then they haven't seen anything. Self-report, when you're asking people to just say what they're doing, that isn't always the best option. We're going to be sneaky. We're going to get around this by using their My Anime List account. In theory, if you randomly allocate your participants into each of these groups, then they should be comparable, because if there's any differences, it should even out. But that isn't always the case. So for example, you might, find that participants in your sitting in a room for 15 minutes group, you know, if you've recruited a bunch of Zoomers that have the attention span of a flea, then they just, 2 minutes in, they're like, I'm bored, I want to leave. And that means that you have what's called selective attrition. So your study has been designed in a way that one experimental group is more likely to leave the study. Ideally, that won't happen. If you've designed your study well, then it won't happen. I think this is a terrible study, so it probably would, but you know, it's a hypothetical. Work with me here. So a hypothesis is just a statement saying like, I expect this to happen when I manipulate my IV. It can be just a general statement. So I expect group 1 to be different from group 2. Non-directional. Do you see what I'm saying here? There's like a direction to the effect that you're seeing. A non-directional hypothesis would look something like exposure to VTubers will lead to different in number of hours of anime watched. And then we've got our directional hypothesis. There can actually be 2 of them because there's 2 potential directions. So in the directional one, we'll say participants exposed to a VTuber will watch a greater number of hours of anime than participants in the control condition. Or participants exposed to a VTuber will watch a lesser number of hours of anime than participants in the control condition. Now I've written it out, hopefully you kind of fundamentally see what the difference is there. But when something is directional, it says what you think the effect will be of the IV on the DV. So you're saying what you think the change in the IV will lead to as an outcome in the DV. Whereas non-directional, you're keeping it vague. You just say that you think that there will be a difference, but you're not saying what that difference is going to be. Now, obviously, if you're writing a hypothesis like this, you don't want to have both of these 2 down here because they are directional in opposite directions. You can't have both. That's not how it works. You need to choose one. I am going to say we will go with this one. They'll watch more anime. There might be some kind of theoretical paradigm explaining this relationship. I can justify it with, so for example, you might say, I think that anime fans who watch VTubers might be more likely to develop a parasocial relationship with the VTuber they're watching, and that means that they will then consume a greater number of hours of the type of content that the VTuber fits into, so anime. The research team asked participant researchers, what do you think will be the direction? So basically they asked them to come up with a hypothesis. They found that on average, the raters correctly predicted the direction and significance of self-confirmatory tests 42% of the time, incorrectly predicted null results 38% of the time, and incorrectly predicted the direction of the findings 20% of the time. That's not great. So that's basically saying that when they asked these scientists whose entire job it is to like understand these kinds of findings and make these kinds of predictions, they found that they only got it right 42% of the time, less than half. Oh dear. Well, I mean, to be fair, there were 4 possible outcomes. So there was correctly predicting the direction. There was incorrectly predicting null results. There was incorrectly predicting the direction, but there was also correctly predicting null results, right? Because they, in theory, they could have found things that didn't replicate and people predicted that they wouldn't and they were right. It seems like that just didn't happen. So I'm not sure what to make of that other than some people maybe to learn a bit more about how forecasting works perhaps. And learn a bit. Read Super Forecasters by Philip Tetlock. That's a good book. The results aren't really convincing, if I'm being honest. Yeah, I'm also slightly concerned by that. You'd think that people that are meant to know what they're talking about, because they've spent literally their entire life researching it, would have a slightly higher chance than 48%. Although it is double what they would get by chance, just because there's 4 possible outcomes. So just like without kind of accounting for what they actually found in the end, you'd expect 25% of them to be in that correctly predicting directional thing. Of course, we now know that 97% of them did actually replicate, so I guess that brings chance level down. But they were trying to make experiments that were not obvious, right? Yeah, so I think they were just basically testing a wide range of different things. The things that they were testing in this study they were new. They were effects that that hadn't really been investigated before. So I don't know, maybe it just reflects scientists' skepticism generally, which in that case, that would be a good thing. On multiple pre-registered criteria, the participants were no better at predicting the outcomes of the mean of highly replicable discoveries presented here than at predicting other less replicable findings from the prior investigation. Oh, that's sad. Well, to be fair, if they didn't know how these studies were being run, then they might have just assumed that they had the same chance of replicating as every other study. And notably, the average accuracy rate of researchers in the first study was nearly identical to the average accuracy among lay people in the second. Oh, I see like 2 triangle symbols and I do think of delta, but maybe I haven't studied enough to know what those means. No, they are delta. They are delta. They're showing the change in the mean between the different surveys. So yeah, the difference between the scientists and the lay people sample. Additionally, the accuracy of predictions for specific findings was significantly associated with the absolute magnitude of the average effect sizes from independent replications. Okay, so the bigger the effect size, the more likely people were to correctly identify that it was going to be replicable. Absolute effect sizes explained 35% of the variance in predictability rates. So the size of the effect that they were finding explained 30% of the variance in predictability rates. 35% of the change that they measured in the predictability rates, how likely people were to correctly predict that the study would be replicable. 35% of that change in that measure was down to the effect sizes of experiment. The findings indicate that the sample of discoveries used here was not of a prima facie different type of content that would yield high replication rates. Did I say that Latin bit right? I've never actually been taught how to say these things, and I just make it up. So I assume it's prima facie, but I might be wrong. That just means that the types of things, the phenomena that they were researching in these studies, they seem to be pretty similar. There doesn't seem to be any immediate difference with any kind of other effect that you might research in psychology. Nor were the content or hypotheses more obvious or predictable than similar findings with low replication rate. It doesn't seem to be in any way like qualitatively different to any kind of social science findings that aren't replicable. It's just, they're just being done more robustly. So fair enough. It's just the same as we've always done it, but better. Cool. Well, now we know about that, we can go back down to the discussion. Our high replication rate thus cannot be attributed to our studies being different in especially replicable topics, designs, or hypotheses. It would also be possible to imagine that we observed higher replicability than other investigations because of the qualities of the researchers involved in this project, such as being better at imagining or discovering new replicable phenomena. Bruh. I mean, not to toot my own horn or anything, but no, fair enough. I mean, the list of people, they are pretty big names, to be fair, especially in the kind of open, robust, reproducible science area. So, you know, there is a chance that they could be better at discovering replicable findings. It has an actual valid point. Although we could be motivated to believe this is a possibility. The principal investigators of this project all have direct experience with their own published findings failing to replicate. Okay, so they're saying we didn't influence it too much because our science used to be rubbish too. I'm not sure that that's a very good argument, to be honest, especially if you're advocating for these kinds of changes in general. You know, it happened to me too is kind of different to like somebody that's churning out those kinds of findings all the time. Maybe they've got a point, I don't know. In this and other research, the participating labs have established practices of making risky predictions, most of which fail to materialise into reliable phenomena. They're just throwing the dice, they're just going out and seeing what happens. If there is an investigator influence on the observed findings, we believe that it's aligned with our interpretation of the present evidence as being due to the adoption of rigor enhancing practices as lab norms rather than individual exceptionality. That kind of reads like does that read like a humble brag to anyone else? It's likely that we observed high replicability because of the rigor enhancing methodological standards adopted in both the original research leading to the discovery and the replication studies. First, rather than using exploratory discoveries as the basis for claiming a finding, all discoveries were subjected to pre-registered self-confirmatory tests. This eliminated inflation of false positives and effect sizes by pre-commitment to research designs and analysis plans. They're actually expanding upon their exploratory findings rather than just saying like, look at this thing I found, isn't it cool, isn't it wacky? This is definitely how people work, which is unfortunately how a lot of science has been done. So they're saying basically we think that the procedures that we're using are just a bit better than the way that science has been done previously. Once a discovery was submitted for a self-confirmatory test, we committed to reporting the outcomes. This eliminated publication bias, which is particularly pernicious when selective reporting of study findings systematically ignores null results. So there's a bit of a tendency, not just in psychology, in like all of the sciences, where if you see an original paper, and you're like, okay, I'm going to replicate it or I'm going to build upon it. I'm going to do something funky with it. And then you do your research and you get to the end and you've got null findings. Yeah, I think that in itself is really, really important information because it tells you where the limits of your theory is. We talked about boundary conditions before. It tells you where your boundary conditions are. But that is not how the scientific system is necessarily set up. To prove that they're good scientists and to get promotions and to get new jobs and all of that jazz and to get money to do more science, you have to get publications. And publications go into journals, which are ultimately basically just for-profit companies that publish the results of scientific findings. And that means that they have their own set of biases that aren't that dissimilar to the mainstream media. They're interested in clicks. They are interested in attention, having something funky that goes viral and then gets more attention and gets more citations, because the more citations that a paper gets, the more influential it is perceived to be by scientists. And then it also inflates a whole bunch of metrics that universities use to assess how important a piece of research is. But those same metrics look good for the journal, which means that if you have the research equivalent of clickbait, like a wacky finding that completely goes against everything that we expect, researchers are more likely to be interested and to build upon it and to cite it. And then the journal's metrics go up. And then everyone thinks, wow, look at how many citations that journal gets. It must be a really good journal. At the end of the day, scientists are also people, and we are completely able to be influenced in exactly the same way as everyone else. Should pre-registration commit you to releasing your data regardless of the result? That's a really good question. So it kind of depends on the data, to be honest. Some of it, I would say yes, absolutely. But some of it, especially in social sciences, you can't necessarily release the data. For example, if we're thinking about a psychology study where you're looking at people's mental health, that is clinical information. It is medical information. It needs to be kept private. It needs to be kept confidential. And even if you've pre-registered your results, you can't then go and post your data publicly for anybody to look through, because there is, even if there's a slim chance, there is still a chance that somebody may be able to work out who that person is. You know, if they've got like a rare diagnosis and also it says where they live in a country and maybe roughly what their income is, then you can work out, well, this person probably lives in this postcode and there's only 3 people with that diagnosis in that postcode, which is really, really dangerous. That's putting people's privacy at risk. So we have a whole bunch of like procedures and policy around how to manage that, when we can share data, when we have to share simulated data. So we run a simulation which takes the descriptive statistics of a data set and kind of infers new data points. It's got the same parameters, but it's a different set of data. And then we can release that instead and say, like, you know, you can rerun the same statistical tests. It will come out with the same results. It's just not the same data anymore. So it protects privacy. Pre-registration in itself at the minute is just a kind of self-governed thing. Scientists pre-register it, and then it is on them to share the outcome of that pre-registration. Unfortunately, it doesn't always happen. In fact, I think there was a study that came out a little while ago that showed it was only about 40% of pre-registrations actually have any follow-up in the form of a publication or in the form of a shared data set or anything. We have preprints as well, which are basically journal articles before they've been peer-reviewed. So that that can be another way to share null results and get them out there, even if a journal won't take them. But to be fair, journals are getting better at accepting null results now. There's no external requirement to share your findings after you've pre-registered. It's just a kind of trust system, which isn't ideal. To be honest, peer review in itself is also a trust system. So there have been quite a few cases that even I've seen, like just in my day-to-day work, where somebody has done a pre-registration and they've said that their research is pre-registered. But when you click on the link and actually go and look at the pre-registration, it basically doesn't say anything. Like, it's so vague. There's no explanation of like how they're generating their variables. There's no explanation of how they're removing outliers. So like, you know, really wacky out their data points that don't really conform with the general trend. They're really easy to manipulate because you can have an outlier that just like totally changes the mean of an entire variable, and then you exclude it, and then suddenly you've got a different mean, and that might change the statistical significance. Sometimes pre-registrations don't even mention them, and it's like, what are you doing with this? In theory, there should be some kind of follow-up on this, I think, but scientists that are working in academia, so like in universities and research institutes, for them, a big part of the appeal of their job is being able to just have freedom, the autonomy that comes with a job. So if there was somebody saying, oh, you need to follow up on your results, well, then they're kind of like, well, I don't want to, and I don't have to. It's not part of my job. Yeah, it's a difficult one. Really, they should, basically. In short, they should. Whether they do is another story. All self-confirmatory tests and replications were conducted with large samples, resulting in relatively precise estimates. Because if you've got a bigger sample, you can be more specific about where your parameters are. That's just a kind of general rule. If you think about things just like randomly moving around all the time, because it kind of averages out basically. That's like such a bad explanation for that, but we'll go with it. We'll go with it. Fourth, each lab was part of the process of both discovering and replicating the findings. This may have motivated teams to be especially careful in both characterizing their methods and carrying out their replications. Yeah, I mean, it turns out if you're being monitored for what you're doing, you actually kind of pay attention to doing it properly. Fifth, if there were essential specialized materials for the experimental design, the discovering lab made them available as supplementary materials. Sharing the original materials should increase understanding and adherence to critical features of the original experimental methodologies. So supplemental materials are like an extra bit that goes onto scientific publications. They contain all of the details that you can't fit into your nicely formatted journal article, because again, journals care about readability and clicks because that's what gets things cited. So all of the boring technical stuff gets yeeted into a different document. But they were sharing that document so that they could actually follow the specifics of how they ran the experiment. We expect that all of these features contribute to improving replicability to varying degrees. Future investigations could manipulate these features to learn more about their causal contributions to replicability. Scientists love to do this thing in their discussion sections where they're like, we did this, but we didn't do this. But in the future, somebody should totally do that. They'll list out things like that. They'll say something that a future study could do. To be honest, it's kind of just a way of like kicking the can down the road. It's like, yeah, I know this needed to be done. I just don't have the funding for it yet. Let me come back to this. It's like putting a bookmark. Or it might be, I think this should probably be done, but I am not super interested in doing it myself. So I'm putting this out here as an idea for somebody else to do. Hint, hint. Even when using rigor enhancing processes, independent replications conducted by other laboratories produced effect sizes that differed from the self-confirmatory effect sizes by larger margins than would be expected due to sampling error alone, producing both larger and smaller effect sizes with a heterogeneity in the order of 0.2 standard deviations. Ooh, that was a bit more technical than I was expecting. Goodness gracious. So this is basically saying that the differences in the replications effect sizes were bigger than they would expect just by chance. So sampling error is basically just differences between different ways of sampling or different groups that you're collecting data from. They were different enough for it to be worrying, although 0.2 standard deviations isn't massive. But yeah, it is enough for it to be of interest for potential future research, I think. The added heterogeneity may be due to ambiguities or imprecise descriptions in the materials provided to replicate in labs, or due to some sample heterogeneities introduced by using varying sampling. So yeah, it could be that they weren't listing out all of their details in the way that they should have done, or it could be that the other lab couldn't replicate it in the way that they wanted to, or it could be that the way that they were finding their participants was just slightly different enough to have an effect. The finding highlights the value of multi-lab replication processes even when the original studies follow rigorous practices. Yeah, because it's showing that there can still be differences even if you're, in theory, doing everything in the way you should. We love scientists working together to answer questions. Turns out it's pretty good. An important question is the constraints on the generalizability of these findings to other research. Our 16 novel findings in social behavioural sciences each involve 2 between subject conditions that could be administered online. All samples for the self-confirmatory tests and replications were drawn from online panels of American adults. These 16 findings do not characterise a representative sample of any methodology or discipline, although they do represent common methodologies, samples, and research questions from the social behavioural sciences. Due to the limited number of participating labs, lab-level variation in the replicability of findings was incalculable. To the extent that labs vary in how they select potential replication targets, the replication rates observed in the present study may not generalise to a broader population of research groups, although, as discussed above, the discovered effects did not differ from similar findings when asking laypeople in a group of researchers to predict them. But what this is saying, roughly, is that they were using labs that were using a certain type of experimental manipulation. So you can manipulate the same thing in so many different ways. With our VTuber example, we've decided to manipulate it by showing a 15-minute clip of a VTuber. There's a lot of other content linked to VTubers that we could use. So we've got the clip, we could use a whole VOD, because obviously clips are just a very small nugget of the VTuber. It doesn't represent their personality. Maybe just the more natural speaking and interaction that you would see on a VOD would be different. Similarly, there's like edited videos. So like YouTube style videos that edit down VODs, for example, that would also be different because that is also a different kind of style of content, right? It kind of shows them in a different way. There is art, there is 3D modeling, there are music videos, so many different types of VTuber content, and all of these might produce a different effect. You know, maybe the art would have a much bigger impact than the music video, because if you like the music videos, you kind of have to already be a fan of idol culture, right? But not every anime fan is a fan of idol culture. The majority of anime fans are like shonen fans, so they wouldn't necessarily be interested in music videos. Whereas the art might be more appealing generally, but then 3D modeling might be appealing to only artists who like anime. Songs might be appealing to everybody regardless if you like anime. There's a whole bunch of like extra hidden details that are represented in all of this. And that's what I meant when I said that psychology and social sciences in general are just a lot more complicated than people give them credit for. There's so many interconnections, connecting concepts, and things that you need to think about when you're developing your experiment. It is actually quite intense. To really get an understanding of this phenomenon that we're researching, you'd have to run all of these, ideally, and maybe you'd also have to vary the length of the video that you're watching, or maybe you would have to change something like the art style of the VTuber, right? You know, if you're using like a Vroid model like mine, maybe it would be very different if you're using a fancy live 2D model. Some of them have really unique art styles. Pumpkin Potion has a really cool art style, but that isn't anime, but she's still a VTuber. So there's a lot of things to consider here, right? And all of these things are things that we need to think about when we are doing our experiments. The studies that they ran as part of these replications, they only represent a very, very small fraction of all of those different potential sources of variance. Yeah, they're basically just saying, this is just a small slither of what this could be potentially. They also mention they're all samples of American adults. So in social sciences, culture plays a really, really big role in how we think and how we act and how we behave. You can find that people respond in literally the exact opposite ways in different circumstances because of the culture they were raised in. And so if we're running this study, we need to think about, who are we actually recruiting? Our population, we've written down anime fans. Are they European? Are they American? Are they Japanese? Are they Southeast Asian? Are they Latin American? There is so many different cultures that you would need to consider. And even within these like quick groups that I wrote down, yeah, there are massive differences. Say, for example, let's go with Japanese because that's the most specific one. Are they from a big city like Tokyo, or are they from the countryside? Does that make a difference in their acceptance of the VTuber? Maybe people from cities would be more willing to accept the VTubers because they're more exposed to anime marketing on a daily basis, whereas folks in the countryside would be a bit more inclined towards the kind of folksy traditional type of mascot, that might also impact our experiment. Hopefully you're starting to realize that there is like so many layers that you need to think about with these. You cannot just assume if you read a paper and it says, we found this effect and it was a big effect, you cannot just assume that it represents everyone, that you will find that effect over and over again, you know, across different groups of people. Even within the same countries, like even within the same regions, there can be big differences due to things like how they were raised or whether they've ever experienced poverty or things like that. These are things that you need to think about. They can also affect the replicability because a large chunk of existing psychological research has been conducted with psychology students. We just go for people that are there. We are using convenient samples rather than the best samples. So that's probably a big reason why things don't replicate. But yeah, these are all things that you need to consider when you're replicating studies. The most obvious area for further testing of the replicability ceiling includes more complex experimental or observational designs, in-person behavioural designs that have intricate staging or unusual protocols, sampling strategies that are more inclusive of the world's population, more heterogeneous conditions including variations in the procedure and time between investigations, and research domains beyond the social behavioural sciences. So that's kind of just saying what I've just said again. You know, there's differences between different groups of people in the world. There you can have a whole bunch of different experimental conditions, like the different types of VTuber content, and then also time between investigations. So if you measure something repeatedly over and over again, right? Let's say you measure the amount of hours of anime, you measure it immediately after they've seen the VTuber. So, you know, the 7 days go past and then you record how much anime they've been watching, and then you do it again in a month. There might be a difference between the 7 days and the month, because what we often see is that the experimental manipulation kind of diminishes over time. So if you run it again and again and again, you'll find the effect size will go down over time. It will eventually kind of hit a plateau, which is just chance level. It's just like the average and it just stays there. The time between your repeated measures is really important. To be fair, I think a lot of traditional STEM subjects, they already do this stuff, right? You know, there's already really, really big collaborations on things like the Human Genome Project and CERN. So they're kind of already doing this. They already bring together lots and lots of scientists in lots of labs to study the same thing. But we're starting to do it in psychology as well, which is really good. And they're also suggesting to this specific protocol that they're using with the pilot study and then the self-replication and then the repeated labs replication. And maybe that would be useful in other domains too. Like maybe chemistry, or especially actually drug development. I bet it would be really helpful with that. That might be a bit more difficult though, because a lot of drug development happens in for-profit labs. But you know, the academic side of it could use it. It's also possible that our computer-administered protocols are inherently more replicable, though prior replication efforts and similar methodologies suggest that this is not the case. And our follow-up investigation found that present findings were no more predictable a priori than other findings with similar methodologies that frequently fail to replicate. So they're questioning whether the types of science that they were doing, which is all kind of like online experiments, whether that would actually also translate into like lab-based studies where you actually bring people into a lab and do things with them. Low replicability or declining effect sizes in social behaviour research are not inevitable. We did not observe declining effect sizes due to idiosyncrasies of different lab practices or different sampling conditions. Whereas prior research demonstrates that such declines can occur, the present research demonstrates that they do not necessarily occur. The encouraging evidence here should empower scientists with confidence that we believe to be rigor-enhancing practices may indeed efficiently generate reliable insights. With the adoption of rigorous research practices such as confirmatory testing, large samples, pre-registration, strong documentation, and fidelity of replication, high replicability is possible, perhaps even likely. Wow. So their conclusion here is basically if we do science a bit slower, but a bit more thoroughly, then we should be able to find more consistent effect sizes. So we skipped over the results, we skipped over the methods. Normally I would say those are the most important bits of a paper, but to be fair, they described their methods and results fairly clearly, even just in the discussion. So I don't think that we desperately need to dig into that in today's journal club. I think that might be something that we can do maybe down the line with papers that are a bit less conceptually heavy. So I think with that, we are done reading the paper. Yay! Congratulations for finishing it along with me. And thank you for all staying and putting up with that. Hooray, yeah, success.